Welcome to our webinar for single donor KILR CD16 effector cells to drive robust and reproducible ADCC and T-cell redirection. Now, as many of you know, therapeutic antibodies achieve their clinical efficacy not only through engaging the target antigen on tumor cells, but also through engaging Fc gamma receptors on effector cells or immune-derived effector cells. So, developers of therapeutic antibodies must assess all possible mechanisms of action for these antibodies, including the possibility of ADCC or antibody-dependent cell cytotoxicity. So for such an MOA, assays that are used in a quality environment need to be MOA-reflective, and therefore an appropriate ADCC assay would be one that would measure target cell killing. But as we all know, such an assay would require the use of human immune cells, primary cells, or an immortalized cell line that is from an immune-derived cell type. And those of you who have worked in this space for a while realize the challenges in using immune effector cells in a quality environment. Firstly, those that use primary cells encounter high donor-to-donor variability in these types of assays. Once you identify a donor that is appropriate for your assay, there are often logistical challenges with getting enough PBMCs or NKs to support the life cycle of your molecule in QC. Finally, one can also observe very low signal-to-background ratios in certain assays with primary cells. So some labs have gotten around these issues with primary cells by resorting to using engineered NK92 cell lines that overexpress CD16, the Fc gamma receptor required for ADCC. However, as many of us know, these particular cell lines are very challenging to grow. Often they're not compatible with many target cell lines, as we'll discuss in a few moments, resulting in high background killing. And then there's the added burden of that they require an expensive license fee to use them in a quality environment. So a reliable source of human effector cells could address some of the challenges that one encounters in using an MOA-reflective ADCC assay for lot relief. That is why we're so excited about the introduction of our single donor-derived engineered effector cells, or our KILR CD16 effector cells. These are primary human cytotoxic T lymphocytes that have been engineered to stably express the V158 variant of CD16. Importantly, they are uniformly manufactured in a quality environment to ensure lot-to-lot consistency. Now, shown at the bottom of this slide are some characteristics obtained by flow that show you the types of cells that are actually in these effector cells. So the CD16 effector cells are a polyclonal population that are 98% CD8 positive. Importantly, 98% of these CD8 positive cells are also positive for CD3, and 99% of the CD8 positive cells are positive for CD16, demonstrating the homogeneous and stable CD16 expression in these cells. Now, these are T cells that mediate killing through their CD16, and shown on the left is the classical model of ADCC, where the NK cell, which expresses the Fc gamma receptor known as CD16, engages the Fc portion of the antibody, whereas the antigen-specific end of the antibody engages the target cell. And this interaction, if you will, with the NK cell drives the release of perforin and granzymes and results in tumor cell lysis. Now, our KILR CD16 effector cells function very similarly to an NK cell, where the CD16 receptor is mediating the interaction with the antigen-specific antibody, which then binds to the target cell and triggers cytokine release from the T cells, resulting in killing. So you may ask, why use T cells rather than NK cells if NK cells are the relevant cell background in normal people? And that is because NK cells have a few problems. NK92 lines often used in the QC labs, are often difficult to grow, as I mentioned earlier. But most importantly, they demonstrate high nonspecific lysis with many cell types. And so an example here is shown on the right, where the NK92 CD16 cells were used to promote ADCC in 2 different cell backgrounds. SCO3s, which are highly resistant to background killing by the NK92, and you can see it's the dark blue line showing very small and low amounts of background killing in this model, whereas ARH77s, which are an hematopoietic model and more typical of many different hematopoietic models, you see very high background killing with the NK92, almost 60% lysis in the absence of antibody. By contrast, what you can see is that the KILR CD16 cells mediate almost no lysis, either in SCO3 or ARH77, in the absence of antibody, and that's shown with the pink bars. So the important element here is that most cells are resistant to killing by the KILR CD16s in the absence of antibody. So these KILR effector cells have been introduced as part of our KILR catalog, if you will, to evaluate ADCC, but they are also a universal effector that can be used in almost any cytotoxicity assay. So shown in this slide is just an example of how one might use them with our KILR system, where the KILR CD16 effector cells are thawed and then rested for at least 24 hours before use in your ADCC assay. And then you would combine them with your target cells that have been opsonized with your antibody and add your effector cells. In the case of our KILR system, our KILR target cells, these are expressing a housekeeping protein that is tagged with the small fragment of our beta-gal reporter system. When the cells are lysed by an ADCC mechanism, you see release of this housekeeping protein into the media, and that release can be detected by addition of our detection reagents, which contain the other portion of our beta-gal reporter and substrate. And this provides a homogeneous readout of protein that is released by, in this case, the ADCC event. Importantly, we show in a few slides from now that you can use these effectors not only with our KILR cells and retroparticles, but you can also use them with your own in-house release assays such as chromium-51 or calcium. So, as I mentioned, we can use these with different cytotoxicity assays, including europium, calcium, and other measures of cell death. These cells have the advantages of eliminating donor variability because they are primary effector cells from a single donor. They're easy to implement in any lab because they are supplied as frozen, ready-to-use cells that require minimal culturing. They're fit for long-term QC testing, as we will demonstrate that they have high lot-to-lot reproducibility. And importantly, they are a relevant measure of ADCC and TCR because they allow you to measure cell killing. So this slide demonstrates some of the data that has been generated to contrast these KILR CD16 effectors with primary PBMCs and NKs. So an example on the left is with the Rituximab model. So this is an anti-CD20 antibody. And the same cell pool can be used to assess anti-CD38 molecules as shown on the right with Daratumumab. So on the left, what you can see is that the KILR effectors at the same E to T that we use for primary NKs generate a much larger, or at least a 2-fold higher signal to background and a higher Emax relative to the NKs, and about a 6-fold increase in signal to background and Emax relative to primary PBMC. On the right, we see a similar story with the Daratumumab, where we're getting roughly 6-fold increase in Emax relative to the PBMCs in a very nice, clean dose-dependent response in both models. Importantly, what we can demonstrate with these cells is that you can use them with a similar performance, consistent performance, post-thaw over a range of at least 10 days. So the data shown here shows the same lot of cells that have been thawed either day 1, day 3, day 7, or day 10, and the same lot of anti-CD20 antibody is used to perform the ADCC experiment. And what you can see in the table on the right is that the EC50s vary by less than 5% RSD over the course of these 10 days, and the Emax is similarly very tight and consistent. This is all performed by a single analyst, but the data are very, very consistent over the course of 10 days. So looking at a different cell model, what we can see in a SCO3 model, which is a HER2 model, trastuzumab, is that when you compare the KILR effectors to either NK92, CD16, or a PBMCs from a V158 homozygote, you still see that the percent RSD between assays, so this is over the course of 3 days, that the RSD with the PBMCs from a V158 homozygote give you an RSD of about 42%. It's slightly better inter-assay values for the NK92 CD16 of less than 20%, but the tightest data is obtained with the KILR CD16 cells with a percent RSD of 9.6%. Importantly, we also see excellent interlot reproducibility with these KILR effector cells. These are 4 different lots that have been manufactured over the course of 2 years, and you see that the percent RSD between lots is 34.7%, whereas looking at the difference between 4 different donors can give you values closer to 60%, which is not uncommon when comparing data from multiple donors, even with the same phenotype or genotype for the F4V158. Almost as important as the interlot data is the reproducibility between analysts. This is an experiment showing 2 different analysts who thawed 2 different vials. Both of them were doing this experiment side by side. And what you can see is excellent inter-analyst CVs, where the percent RSD across the 4 vials is 18.3% for EC50, 18.6% for signal to background, and in Emax it's 11.1%. So very good precision that should allow these cells to be useful in a quality environment. So taking that data, we are going to share now a qualification study with our KILR effectors that was performed by our colleagues at Sartorius Dedham. And this is just an example of the study design for this particular, with this group, where they were using an in-house ADCC assay and evaluating Rituximab. And they were evaluating, they used this particular study design shown in the table here, where they were assessing accuracy, intermediate precision, linearity, range, and specificity over the course of 3 days with 2 different analysts, and over the range of 50 to 200%. So this is an example of the qualification results looking at the 100% sample between 2 operators. So the first 2 graphs, the top 2 graphs, are showing data from 2 different operators using the same vial of cells, and you see that there is excellent concordance between the 2. And the bottom graph is showing a second vial tested by operator B, or the second operator. And what you can see is that there is excellent precision, only 11% CV between the measurements of 100%, demonstrating that the assay has adequate precision for lot release application. And this is another example of data from this qualification study where they were evaluating the 80% and the 125% samples. And again, what you see is the excellent accuracy in the table at the bottom of the graph. where there was a less than 2% difference in the 80% concentration obtained with these cells. So the overall qualification results are shown here in this slide. And again, you see the concentration range from 50 to 200%, and that the level of inaccuracy is quite low. Importantly, there appears to be no bias in the assay because there is no segment where the inaccuracy is popping up. What you can see is that any levels of inaccuracy are really towards the end, the 50 to 200%, the ends of the range, which is fairly typical for most assays. And you can see that the same data, when they're plotted for dilutional linearity, give an excellent R-squared value of 0.9989, indicating that this assay is suitable for and accurate for use in potency or lot release. So our colleagues at Sartorius Sedman, Catriona Thompson, and Lisa Blackwood, who provided us with data, concluded that these KILR CD16 effector cells demonstrate a good alternative to isolated PBMC preparations from blood, which is what they typically use in this assay, and that they can be used from clone selection and process development to comparability studies as well as lot release assays. I would like to point out that if you'd like to hear more about this study, we will have our colleagues sharing this data in a webinar coming soon, later this fall. Finally, I'd like to share one more example of an application that can be used with these cells, which is a T-cell redirection application. So we chose for this study to work with the BiTE molecule blinatumomab, which is a bispecific antibody that engages the CD19 antigen on B cells on one end of the antibody, and on the other end, or the other arm, it engages CD3 on the surface of the T cell. And this antibody then serves as a synapse, if you will, between the tumor cell and the T cell, and mediates lysis of the tumor cell. And we can detect this lysis by using, again, our KILR assay system, where we're measuring release of our modified reporter protein into the media and detecting the release of that into the media with our detection reagent. So in this slide, we contrast the kinetics of this response to blinatumomab in both pan-T cells and with our KILR cells, our KILR CD16 effectors. So the data shown here are the kinetics with pan-T cells. And what you can see is in the absence of effectors, we see no killing of the cells. But when you introduce the effectors at a ratio of 10:1 and incubate for 24 hours, you see a robust dose-dependent killing of our KILR Raji cells, which are positive for CD19. Similarly, when we perform the same experiment with our KILR CD16 effectors, you can see that the kinetics are much faster, and we have a nice dose-dependent response that achieves a similar Emax to the 24-hour data that we achieved with the pan-T cells. We achieve this with 6 hours with our KILR CD16 effectors. So importantly, the EC50 that we observed in both of these assays with either the pan-T cells or the KILR CD16 effectors is within the range reported for blinatumomab in the BLA submitted to the FDA. So in conclusion, our KILR CD16 effectors drive robust and reproducible performance in your cytotoxicity assays, either with our target cells or your own in-house ADCC assays. They are single donor-derived primary human effector cells that eliminate the donor variability from your ADCC assays and allow high reproducibility. They are effector cells that have been engineered to stably express CD16, so they are a biologically relevant measure of ADCC. Importantly, we have high lot-to-lot reproducibility that enables the long-term support of your QC lot release bioassays. They are cryopreserved, ready-to-use cells that need at least a 24-hour rest before you can use them. But the protocol is very QC-friendly and easy to adopt in any lab. And they produce a higher signal-to-background ratio than most primary cells, which leads to improved performance compared to primary PBMCs or NKs, enabling their use in loss-of-leaf assays. So the cells can be obtained either as a 1 vial or 5 vial kit. And we also provide the cell culture media and the IL-2 as consumables that you can purchase with the cell. And these come with a certificate of analysis and user manual that allows you to get your assays up and running very rapidly. So if you have questions related to these killer cells, I would point you or direct you to our tech support team, who is available and ready to help you get these assays up and going. And if you're interested in any more information, please visit our website at discoverx.com/killer effectors. I want to thank you for your attention and for attending our webinar, and I look forward to having the opportunity to share more information about these cells in the near future. Thank you.